Okay, so let's begin now with our first subject. So it's about discovering and seeing where we stand at the discovery of the gene. This work is taking place in the hospital. We've split this into three pillars. The first part is about genetics and the METAR gene. Then we'll be talking about the phenotypes associated with this disorder. And then a third pillar on the clinical research of the disorder. Now, the syndrome. Let's go over the genetics of this. Each person is made up of 70 billion cells, roughly. And in the nucleus of those cells, you have the DNA or the genome, which is really our genetic makeup. So it's like a recipe book that tells the body how to function. This DNA is unique to each person and made up of 46 chromosomes, and we'll be talking about those later. This DNA is made up of 4 letters, the nucleotides A, T, C, and G, that go in pairs. And together in the genome, there are 3 billion pairs of letters. These nucleotides, when you bring them together, make up genes, and they are like recipes. We have roughly 20,000 recipes in our genetic codes, 20,000 genes. Now, these genes are made up of different parts. We have the introns and the exons. You've probably heard of the exome, which is all of the exons that are free, and that exome makes up 1% only of the genome. It's only a tiny part of our genetic makeup, our genetic code, and yet it's in this part that most of the modifications or variations appear that are the beginning of all of these pathologies. Now, the role of these genes, or part of their role, is to make proteins so that the organism can function. And these proteins have different roles. They make the heart work, they give indications on the weight, on the size, get the bones to work. It's a little bit like a kind of telling instructions for the proteins on telling the body how to work. Now, the link between two genomes is identical to 99%, but the difference is called a variation. Before we used to call it a mutation, but now we call it a variation, and that means that everybody is unique and different. Now, this percentage represents 45 million variations in relation to the basic genome. And these variations, for the most part, have no impact or can generate different eye colors, hair colors, size, etc. So only very rarely can these variations generate a disorder, a cell disorder or dysfunction, when they impact critical parts of the genetic code and generate genetic disorders. These modifications can happen in 2 different places, either on the chromosome. So you can see a missing part of a chromosome, a deletion, or in the nucleotides, in the genes, there can be modifications of the letters, either extra letters or letters changing, or some that have been deleted. For the most part, these variations are inherited from our parents. But we do have 50 to 100 new variations in relation to your parents. And that's normal, that's natural, if you like, these variations in relation to our parents' genes. And they mostly don't have any impact and don't generate any pathologies. But sometimes, very rarely, they do. And most of the time, it's a problem with the cell generation. That either happens when the sperm is being made, when the egg cells are being made, or when the divisions of the embryo in the chromosomes happen right at the beginning. The METAR disorder is on chromosome 2, as you know, right at the end of chromosome number 2. Now, this gene has a role. It produces a protein with the same name, the methyl protein, and that goes through various different stages. This protein has been modelized by the American team in 3D, and you can see it here on screen. It's made up of different parts, represented by different colors here. One of the parts that's very important is the blue parts, these 6 different parts, the zinc fingers, if you like, and they produce a pocket which attaches itself to different parts of the genetic code to generate the role of the métal gene. Now, this is a 2D representation of that protein, and we can see that the zinc finger is part of that 2D protein. And we're going to focus on the second and third domains or fields of the zinc fingers because they are the ones that are impacted in the disorder or the syndrome that we're dealing with. I won't go too much into detail on the role of the métal gene because the American and German teams will be doing that. But I want you to see it as a conductor. It's like a conductor of an orchestra. It's a gene that's going to be conducting hundreds of genes. And the main task is to turn immature neurons into mature neurons. So that gene is going to, thanks to its protein, connect to different parts of the genetic code and play that role, turn that immature neuron into a mature one. So be careful of the name of the gene. You mustn't get it confused with the myelin production because there is, it has no role in that process. That's what surrounds the neurons. And so MIT1 is totally different, and it's not the MIT1 gene. So they are 2 different things to not confuse. Now, we all have 2 copies of the métal gene, one from our father, one from our mother. You need 2 copies, actually, so that the functions can be respected. Now, in the métal disorder, there's a problem with one of the copies, a dysfunction. So only one copy will actually be a functioning one.
Ça va revenir. Parfait. Et on ne peut pas enlever la petite barre? Non.
Can we take the little bar off? No?
Donc, dans ce syndrome, on va avoir une seule copie qui va être fonctionnelle.
So, with this disorder, only one copy is functioning, and that's a specific mechanism called haploinsufficiency. The only functioning copy is not enough to fulfill its role, in theory.
Donc, ce syndrome, il se transmet sur un mode
Now,
autosomique dominant.
this disorder is transmitted in a dominant autosomic way. That means it affects girls as much as boys, and dominant means that only one modified copy is enough to generate symptoms and the pathology. In most of the genetic code modifications are de novo, only on that particular individual, not on the parent's code, but transmission onto their descendants is a 50% one. Either the patient transmits the modified copy or another copy. Now, as far as the mechanisms go, there are 2 different molecular mechanisms. Either there's deletion of the chromosome, the missing part, so the chromosome deletion either takes part of the métal gene away, a fragment, or the whole gene. The second mechanism at play are punctual or random variations, so missing letters. We mentioned that earlier. And as far as these variations go, these genetic code modifications, there are 2 different ones. But the false ones and the truncated ones. The false ones is an amino acid that's abnormal. So the protein will have a specific form because there is a random problem. But the truncated one stops the gene, it's detected by the cell, and the protein is not made. and there will be no protein, quite simply. And for a number of years, we've wondered whether patients with a deletion, a missing fragment of their chromosome, had the same symptoms, the same phenotype as other patients that had missing letters in their nucleotides. Today, we know that they have the same phenotype. Of course, each person has their own symptoms that are different to other patients, but there is no big variation between their genetic codes. This gene was described for the first time in 2011 by a team that had 6 patients. So they all had gene deletion, and these patients had intellectual disorders, behavioral disorders, obesity or weight problems, and one of them had epilepsy. So 2011, that's not a long time ago, but since 2011, we've been lucky enough to see a lot of papers published, clinical papers or molecular papers. That has enabled us to improve our knowledge, increase our knowledge on these patients that have these random variations. Now, in 2019, when we began here in Rouen at the hospital to look at this syndrome, there were only 14 patients covered in the medical studies. Only 14 had been described, so we only had data for 14 patients. And we already knew then that these patients had neurodevelopmental disorders, problems learning to speak and walk. They were mentally retarded, they had behavioral issues, they had weight issues, epilepsy for some of them, and sometimes even problems with the brain in MRI scans, and many, many things. But in 2019, even though we already had some idea of the phenotype, the syndrome was not very well described. We didn't have good data and not very well phenotyped. And on a clinical and molecular level, we had uncertainties. And that's why we wanted to focus on this disorder. And the main objective was to try and better describe the phenotype, to try and update the data, try and identify new symptoms maybe even, and take a look at whether there was a link between the types of genetic variation, the types of modification on the nucleotides, and the phenotype. And to do that, we set up a team, and they worked for several years. We got in touch with professionals from all around the world, but mainly in France, Belgium, Luxembourg, and the US, and we included 14 patients, as many girls as boys, with a median age of 11. I'll now talk to you about the phenotype that we have come to know in 2025. That's the fruit of all of this data. But what you need to remember is that antenatal, during pregnancy, there are no specific symptoms linked to this disorder, to this syndrome. Of course, there can be malformations, there can be maternal symptoms, but they aren't necessarily linked to this problem. And today, we don't know of any symptoms that mothers-to-be may have. But when at birth, there are no specific problems with growth. Babies are born at full term. They have normal sizes and weights, but 15% of the patients have hypotonia at birth, so restricted movement. And in 100% of cases, these patients go on to have problems walking and speaking in relation to other children, and specifically in speaking. But before that, there are motor problems, so they find it hard to sit up. They do that at 11 months instead of 9, and they walk more like at 18 months rather than 12. And those motor problems also can be seen in the finer motor problems. And then there's that general hypotonia that some patients see for several years. Now, on a language level, there is a delay. First words are said at roughly 2.5 years, and first sentences at 5 years. That can vary. Some patients find it harder than others, but we have these syntax and phonological problems in most patients, and we'll be going into that a little more later. But this neurodevelopmental disorder then develops into cognitive problems in most cases, and they are very variable. Patients sometimes don't have too many problems and others much more. Some patients who don't have mental retardation, according to the international norms, may have learning difficulties. So, for instance, all of the dyslexia, dyspraxia, etc. But there are also visual and attention deficits, and they can also impact the problems, the cognitive problems already present. And most patients need special sessions with all kinds of practitioners to help them. And schooling needs to be adapted, but that can be different from one patient to another. Some only need an extra adult standing with them and helping them a little bit in a normal classroom, but other patients might need to have to go to special needs classes of different kinds. So care can vary a lot, and it depends on the patient's symptoms. Most of the time they have pediatricians following them, sometimes endocrinologists, psychiatrists, ophthalmologists, geneticians, at least once for the diagnosis. And then they have a paramedical care pathway that can evolve with time and according to the needs of the child. Now, as far as behavior goes, we have noticed that the disorders are constant, but can take the form of two different types. Some patients have classic autistic problems. Others have partly autistic problems without being completely on the spectrum. There are intolerance to frustration, sometimes aggressiveness, which can be auto or hetero, sometimes attention deficit, anxiety, hyperactivity, etc. Now, all of these disorders can develop further in life, and those are things that we don't have enough information on. What impacts that change, and they can either be on their own or together, conglomerate. And usually it does have an impact on the patient and on the family. Now, on a neurological level, some patients have epilepsy. We'll be talking about that this afternoon, but we have different kinds of epilepsy. There's no specific kinds of seizures according to what we can see. The severity varies. Mostly it's an epilepsy that starts very early on in the neonatal or early years, and depends on the kind of seizure, what kind of treatment it has. So treatment does normally work, but that depends on the patient.
As far as the neurological issues are concerned, there is no regression, there is no loss of intellectual power or motor capacities. However, this data is limited, and that is something we have not yet observed. No muscle disorder either associated to the gene. However, many patients will show modal hypotonia with coordination disorders. They can be clumsy or they have balance issues. Brain MRIs have been carried out on a number of patients. In 28% of the cases there were abnormalities. However, the abnormalities were not specifically related to the syndrome. We could not find any symptoms in the patients who showed these abnormalities. And most of the time the abnormalities were not really serious and did not warrant subsequent monitoring. So no specific image was identified for this particular disease. Regarding eating habits, many patients, 60% of the patients were overweight or even obese, but this may vary in time, and the patients sometimes will change their weight curve depending on the age, but when they put on weight they did so very early on, in average at 3.5 years of age, and that's very important. This is something that we're going to look into later. And besides the fact that they were overweight or obese, they also had behavioral disorders such as difficulty managing society feeling, hyperphagia. Children who eat very fast are definitely obsessed by food. In a more infrequent way, we found the people with the opposite phenotype, people who had trouble putting on weight, initially, very early on in life, with troubles sucking on the bottle, or they had to undergo a hypercaloric diet in the early postnatal period. This is rare data, but it can happen. Then we have eye-related disorders, 30% of the cases, not serious, refraction, strabism, also sleep disorders, 33%, which can be have an impact on the patient's quality of life and the family's quality of life. Children who will not go to sleep will wake up at night, and sometimes in the background there may be endocrine disorders such as thyroiditis, microcephaly, which has been described but doesn't really happen very often. We then went on to ask ourselves, people with this syndrome, could they have also show signs in the face that might evoke the MITL syndrome because they had developmental retardations, as happens in other diseases. So we worked on the face-to-gene software. It's a machine learning software that uses the photographs published in the literature in order to give us a typical face where we can find signs that are quite evocative of the disease. And these signs can be found in some patients. We have eyes that are sunken and almond-shaped, and the nostrils can be tilted forward, and sometimes there are signs that can be related to the obesity. Now, the symptoms are not constant and consistent for all the patients. Sometimes you will observe them in different syndromes. It's very difficult to make a diagnosis when you see the child for the first time for neurodevelopmental retardation. It's very difficult to think about that syndrome first, only based on the signs seen in the face. We then went on to look at the molecular changes, nucleotide changes in the gene. There are two types of variations. There are protein truncating variations and missense variations. Now, in most cases, the missense variations are the ones that change the protein shape. These variations are clustered. They will cluster together in a very specific place, the second and third domain in zinc finger domains, and some patients show variations that can be found outside of those domains, but that's rare. Whereas the truncating variations that break the gene reading and will lead to the absence of some proteins, they can be found all along the protein. We wondered whether the patients who showed missense variations clustered in the middle of the zinc finger domain had the same phenotype as other patients who had truncating variations. We compared a number of clinical criteria and we could find no difference. The patients had suffered from the same symptoms. There was no phenotype-related variation, no genotype-phenotype variation between the two types of mutations. The assumption, the hypothesis could be that missense variations that change the conformation may be at the origin of a protein. Protein is there, but it's not active. And this has the same consequences as if the protein were missing altogether. Then we looked at the patient's phenotype and compared them when people had a missense variation outside of the zinc finger domain. Unfortunately, we only had 3 such patients, and we could not make any show any difference. So to conclude on the phenotype, the patients showing MITL syndrome have neurodevelopmental retardation issues, sometimes psychiatric disorders, sometimes sleep disorders, sometimes eating disorders. They eat fast or they put on weight or become obese. Sometimes they have morphological specificity and behavioral disorders. To conclude, I would like to cover the clinical research associated with the MITL syndrome. The fundamental research will be covered later by other speakers. The MITL syndrome is a rare disease. The European definition is the following. It's a disease that will affect less than 1 person on 10,000. We think we have 70 patients in France affected by the disorder. We don't have a database that will help us list all the patients, both in France and abroad. Maybe 1,000 patients across the world, but again, again, we don't have enough data. As you probably have surmised, this syndrome is a rare disease and we are facing many challenges. First challenge, because it is a rare disease, the patient cohorts are very limited, very few patients in France and abroad, and therefore biological clinical data is limited. If we want to acquire more knowledge, more in-depth knowledge, we have to organize national and international cohorts. This is the first challenge, how to recruit patients from across the world. Second challenge, the data that can be gathered anywhere is unnecessarily comparable because every country has standards regarding overweight and obese patients. The standards are different. It's very difficult to compare the data. This is essential. We need to find a way to perform rigorous scientific studies so that we can interpret the data with the aim of being able to improve the knowledge, but also improve the management of the patients. We can split MITL syndrome research in 3 stages. It would be the same for other syndromes. Phenotyping first. Phenotyping means characterizing everything that can be described in terms of symptoms, clinically, but also molecular, for molecular purposes, variations in symptoms. Second stage, we need to find a model, understand the biological models, the cellular models, the animal models, and understand what pathways go awry and when. We focused in Rouen on stage number 1, phenotyping, and stages 2 and 3 are left to the German and American colleagues. Stage 3 is the one we're all pointing towards, how to find therapeutic targets that are specific to the syndrome. So why is it important to characterize at best the phenotype and find the symptoms? Well, because we need to improve the way we attend to those patients on a daily basis. This syndrome is extremely rare, and therefore most healthcare professionals who attend to those children are not familiar with the syndrome. Maybe they haven't even heard about it. The disease was described recently for the first time. It's essential that we train healthcare professionals on this disease and that we come up with follow-up recommendations both for you and for the healthcare professionals. Secondly, based on the clinical characterization, we need to try and understand the underlying biological laws and understand the natural course of the disease so that we can anticipate on some symptoms that could appear later in life. And also, if we can characterize the symptoms in a more efficient way, then we will be able to describe assessment criteria looking for future therapies. We will know exactly what we are looking for, what we're trying to regulate once therapies are available. For instance, if we want to use therapy against epileptic, what is it that we want to control? No more seizures or no more seizures on the electroencephalogram? Do we want to decrease food obsession for food? And it's very important that we understand what exactly we are looking for so that we can adjust the therapies. So we conduct studies, observational studies, interventional studies that may have a bigger impact. We ask people to fill in questionnaires. We ask patients to come back to the center to do a checkup, to collect real-life data so that we can have enough data at a given time, or if we want to go over the natural course of the disease in a more exhaustive way. In this syndrome, there are different issues which require that we perform clinical research. And in Rouen, we focused on 2 subjects, obesity and eating disorders. And this will be covered by my colleagues later during the meeting. We will show you some slides with the questionnaire that we sent to a number of French-speaking families. And then the second research project on Rouen regards the neurodevelopmental disorder. Now that the project was started in February, it went on for 2 months. I will explain. And the goal was to improve knowledge in 3 orientations: language, prosody, voice, speech, and cognition and behavior. The goal was to acquire a better understanding of the symptoms, to try and maybe identify specific profiles so that we can adjust the way we manage those patients, and maybe we can find other therapies. For this project, we asked the French-speaking families, because everything was conducted in French, so we had to limit ourselves to French-speaking families, and unfortunately no one from abroad. We asked them to come to the university, the teaching hospital in Rouen for a couple of days to perform 4 interviews, assessment, 1 genetic assessment in order to go over the history, previous therapies, the patient's history, how the patient was managed, and then depending on the patients we conducted a oral speech assessment. This was done by Constance and Laura, and they will introduce their work this afternoon. Prosody was also assessed, how the patient speaks. And finally, Cécile and Sophie and Hugo looked at the sleeping patterns, and they will deliver a presentation this afternoon. Now, the protocol lasted 1 day, and thanks to the association, we had funding to provide transportation and accommodation for the families, and we saw 1 or 2 patients per day for 2 months. 22 families came to the teaching hospital. We were very lucky. We don't have the results yet. This will be the project for this year when we interpret the results. And just to close, there's another way to gather data, and that's what Valérie was saying earlier. The databases that will be filled in by the families. Two databases that we're in contact with, the French one, Genida, and the American Simon Searchlight. Initially, it was dedicated to research on autism, and the Gemila platform was started by the Strasbourg University. The families send the data based on questionnaire prepared by doctors, and we also worked to adjust the questionnaires for the MITL1 syndrome. And more recently, we restarted the work because we wanted the platform to be fed by the families. And this is international recruitment. It's accessible in many different languages, so the questionnaire can be filled in by many different people speaking different languages. And therefore, we gather results which will be then correlated to the results we obtained with our own studies that we conducted here in France, in Rouen. That's a slide on the Simons Foundation in the States. They offer recommendations for patient management and for the families. There are mini guides for each syndrome, and based on the questionnaire, we gather data from the families, and then we can offer to the family and scientific community all the results we have gathered. I will close with this slide. I think we're all holding hands around the MITL syndrome. Clinicians, biological researchers, basic research scientists, and we're all connected to the patients, to their families. We want to work together. We want to improve our knowledge so that we can subsequently improve the patient's daily life and their family's quality of life as well. I would like to thank all the patients, their families, all the families who came to participate in the research project. They filled in questionnaires, they filled in many questionnaires. The association, because we now have been in connection for many years, all the co-workers, doctors, scientists, researchers who worked with us, and the program unit that has been coordinating the projects, the med school that is hosting our work, and all the professionals who worked on this project on a daily basis. Thank you very much.